And today we're gonna do a grand stain. Grand stain is a pretty long lab, so we won't have time to do the question from last week. Hopefully we can get it done in about an hour, grand stain. And the last week we did symbol stain. Symbol stain is use one dye, and grand stain is a differential stain, meaning we use two dyes, different colors, so called differential. And let's turn to your book of grand stain, chapter 37 on page 195. So we talk about the grand stain theory, theoretical background, right? Remember in the lecture? Anybody remind me about the grand stain theory?
Doesn't it help like tell the difference in between like different organisms?
Yeah. The grand stain can tell gram negative versus gram positive. The two big families of bacteria, very different, right? And about the stain itself, and with two dyes, one is called a crystal violet, that stain the cell, both type cells, purple. This is like a symbol stain. So the first step is symbol stain, and the second step is decolorization. Use alcohol. Use 90% alcohol decolorize, and only the gram-positive cells can retain the dye, and the gram-negative eventually will be decolorized, and will be no color. Then we need to use a counter stain to bring the gram-negative cells into a color, back to the color, with siphoning, which is the pink color. So in the end, the gram-positive cells will be purple color, and gram-negative cells will be pink color. In between, we use iodine. Okay, so after the primary stain with a crystal violet, you with iodine. Iodine will immobilize the dye into the cells, so that they cannot be easily decolorized by alcohol for the gram-positive bacteria. But gram-negative still they cannot retain the dye. Anybody can remember the difference between gram-positive, gram-negative cell walls?
One of them has something that starts with a P. I can't remember. It's like something.
Peptidoglycan, right?
Yeah.
And what's the difference between the two types of cells? Anyone? They all look similar. I mean, by structure, gram-positive cells will have multiple layers. Remember, right? We talk about 29 to 30 different layers, and gram-negative cell only have one layer, sandwiched by outer membrane and the inner membrane. So that's why there's a single layer of cell wall cannot retain the dye. Eventually, it can be easily decolorized by alcohol, right? Okay, now let's turn to the page on this textbook, the lab manual, 200. And we're going to follow basically follow the procedure of grand stain operation. And first of all, we have you have this slide, right? Take one of the McCarthy slide, make three portions, three areas. You can use a marker on this side and put a mark on this side and put a cell on this side, not same side, right? So the marker won't get messed up with your staining. Then like we did the symbol stain, you add a loop of water in the three areas. I can see here, one loop of water over here, and 2 and 3, right? In the first session, you take a cell, take E. coli, from a plate with a loop. And remember, don't scrap too many cells. Make some. You can see if you can see that some cells, some colony on the surface of the loop, then it'll be enough. Don't scrap too much, and mix it with the water, and then make smear like you did last time. And on this side, you take Streptococcus epidermidis, which is a gram-positive control, and E. coli is a gram-negative control. So we have two control, one on this side and one on this side. And then in the middle, you're gonna take your chick cells together with the bacteria, normal microbiota in your oral cavity. You take like you did last time, right? Make smear over here. And then you're going to dry on the top of a flame. Flame here, you go a little bit higher. I don't want to overheat, overcook, I don't want to barbecue yourself, right? You want to heat it up when you see the water is gone, evaporate, and that's it. Don't never overheat. Then this is step 1, and step 2, you cover your smear with ultraviolet. Just a few drops. Make sure you cover the whole smear, and let it stay for about a minute. And then you wash it out with tap water, which is pretty. Let me use the tap water. Okay. And this step is also very critical. Remember, don't wash this with too much force. So you wash like this, all the cell will be gone, right? Yes, make sure you have a slow, gentle flowing of water or dripping. Yeah, this will be safe. You have to wash it like this. You can wash the dye, go and dye to the sink, so it'll be okay. And wash it, keep it running, so it's okay. Then use a tray. So I have a tray here. Yeah, use a tray. And place this on a tray like this. Okay. Go back, see everything here. Okay, on the tray. Then you cover this with iodine. First, crystal violet 1 minute, iodine 1 minute, and wash out iodine. Then you decolorize with alcohol. So we have alcohol here. I'm going to bring alcohol. It's almost pure alcohol. It's 90% or 95% alcohol, and it's flammable. So when you play with alcohol, don't operate near the flame. Okay, this is the hood, the chemical hood. So we're going to operate the chemical hood and take this alcohol. Yeah, bring your slide here and add a few drops of alcohol to cover the whole smear. Remember, at this time, the decoloration time is very critical. If you over decolorized your slide, then there'll be all the gram-positive cells turn negative, everything will turn pink. If you under decolorize your slide, then it's the opposite. All the cells turn gram-positive, all the purple cells, even gram-positive cells, gram-negative will turn purple. It's called artifacts. Okay, and then when you're done with so the time based on my advice here is you have slide here, you ready to add alcohol, and you count about 10 seconds. You count, so you count 1 through 10, but when you count number 7 or 8, you go back to your bench and start wash out. Then eventually it will be 10 seconds, around 10 seconds, right? And wash out the alcohol. Then you do a counter stain by siphoning. You cover slide with siphoning. let it stay for 1 minute, then you wash it out, and then you that's the last thing here. Okay. You can operate the whole thing with this clip, slide clip, so there'll be a stay away from the surface of the tray. The floor of the tray is very dirty as here. And the last step, when you're done, you remove your slide and place on the paper towel with the face up. So the cell on this side. Don't do this way, right? On the top with the face up position. So that the bottom part of the slide will be dry out, and then let the air dry until you see a residue of water. Meaning you can have probably 50% of air dry. And then you put a cover slip on, 3 cup slip. Okay, three here, 1, 2, 3, and look at the mask scope. So when you start the scope, when you put a mask scope, start with a low power lens to bring your smear into your vision, into the field of your vision. And then you can zoom in with a high power, eventually see cells. First, look at the 2 controls. Gram-positive cells should be purple, and gram-negative cells should be pink. If the 2 controls are not as expected, meaning your procedure is not correct, then you go to see your cells, the cells of normal flora, which is very complicated, and it's a mixture of gram-positive but negative with different type of shape together with your human cells. Right, buddy? And here over there we have E. coli plates and S. epidermidis plate, and so we have each one of you have your own plate, and slides over there, and cover slip, everything, and you see on the table, you see 3 bottles, right, in the front of you, the solutions of quick-ballot, iodine, and saccharine, right? Any questions? Okay, let's go ahead, and we just only 15 minutes past. Let's go ahead and get it down in the next 40 minutes.